Hello and welcome students. This is our second video in our first chapter, Reproduction in Organisms, right? So we will continue with the different methods of asexual reproduction. So today we are going to see fragmentation. What is fragmentation? It is the process of breaking a piece of organism followed by cell division or mitosis, okay? So in fragmentation, an organism is broken down into many pieces and each broken part develops into a new individual. So in Spirogyra, fragmentation takes place and Spirogyra splits into several pieces and each of those pieces will be able to grow into new Spirogyra. So besides Spirogyra, there are very common examples like cyanobacteria, molds and lichens. They also undergo fragmentation and other forms of algae. Okay, Spirogyra is an algae. Several other species of algae also undergo fragmentation, right? The fragmentation capacity of an organism depends on the complexity of the organism. So very complex organisms cannot undergo fragmentation. So fragmentation is only a process that involves less complex organisms or primitive organisms. They should not have any complex organelle structures. Okay. So sometimes fragmentation may be intentional or it may occur naturally or it may occur due to predators, due to the presence of predators. So fragmentation in Spirogyra, you can see a diagram of that. Spirogyra is an algae which consists of long thread-like structures, and in each thread you can see a series of cells. Okay, so each thread in Spirogyra can split into several pieces due to fragmentation, and each of those fragments will start growing cells longitudinally. It will start dividing. Due to cell division only, it increases in length, and each of those strands will increase in length and form new Spirogyra. So next is regeneration. Regeneration is a modified form of fragmentation and it is much more complex. Regeneration occurs in much more complex organisms, and the speciality of regeneration is that the number of pieces depends on specific parts of the organism. Okay, so regeneration does not take place randomly. The splitting of the organism takes place at specific parts. Okay, it is known as a process that makes genomes, cell organs, organisms and ecosystems resistant after alterations or damage. So generally regeneration takes place to repair the damage that has happened to organisms. So in commonly we can see in lizards how regeneration takes place. In lizards, regeneration is not used as a method of reproduction, but when a predator is nearby, a lizard will cut off its tail. It will leave off its tail to distract the predator, and later it will be able to grow a new tail, right? So therefore the growth of the new tail shows regeneration in lizard, and in lizard if any other parts are being cut, like the other different parts of the lizard cannot be regenerated. Okay, only the tail gets cut off and only the tail can undergo regeneration. So regeneration is specific to organs, whereas fragmentation is very random, right? So all species that live on earth can regenerate, but only a few species use it as a method of asexual reproduction, thus producing new individuals for their body parts. Especially flat planar worms, they use regeneration as a method of reproduction. They have highly adapted to regeneration capabilities and they use it for asexual reproduction. Among vertebrates, tail amphibians, salamanders and newts and certain lizards, geckos, are highly adapted to regenerate their limbs, tails, jaws, eyes and certain internal organs. In humans, we can regenerate only our liver. Okay, only the liver has the capability to regenerate, that too to a limited capacity only, right? As they are more complex multicellular organisms, they cannot use regeneration to reproduce or as an asexual reproduction method. What is the meaning for that? Complex multicellular organisms will have different organ structures and organ systems. So therefore, if any of these portions are damaged, it will be very difficult to regenerate such a complex system, right? So complex organisms do not have the capacity to regenerate. They can regenerate only individual organs. So even lizards, if you've seen, they can regenerate only the tail, which is a simple structure. The other parts of the body which have complex structures cannot be regenerated. Even regeneration is seen in starfish. if any of the arm of any one of the arms of the starfish is cut off, then that arm will be able to grow the new starfish again. So even starfishes use regeneration as a method of asexual reproduction. So you can see that in the diagram, how the starfish has regenerated. So the single arm that has been cut off can even grow the remaining parts of the body, and a new starfish is born, and the remaining part of the body also forms a new arm, right? In lizards, in wall lizards, you can see how the tail has got cut off, and slowly, after many weeks of time, it grows a new tail. So this is the process of regeneration. Let us now see the differences between fragmentation and regeneration. The first point, fragmentation takes place in multicellular organisms having simple body organization, whereas regeneration takes place in fully differentiated multicellular organisms having complex body organization. Each and every piece of body produces a new organism, but in regeneration, each and every piece of the body may not develop a new organism, only specific parts regenerate. In fragmentation, no specialized cells are involved, whereas in regeneration, specialized cells proliferate and form a mass of cells. Each cell of mass differentiate to new cells and tissues. So the process of differentiation is seen in regeneration, whereas in fragmentation, it is a more simpler process. So the last method of asexual reproduction is sporulation or spore formation. So this takes place mostly in fungi. Spores are single-celled minute propagules produced by several organisms, especially fungi, which are able to develop into a complete organism, right? So spores are specially formed structures, and they can spread to long distances through air or through wind, and they can grow into individual organisms. So spores form a successful method of reproduction for fungi, and they help the fungi to develop and to grow, to reproduce, and to spread out in more amounts. Okay, so spores can be exogenous or endogenous, right? So spore formation we can see in the bread fungi, then rhizopus, and we can see even in the other different forms of fungi, right? So here what happens is a structure called sporangium is formed. You can see that in the diagram. A sporangium is the spore case inside which the spores develop, and later when it has matured, it will break open and release the spores. The advantages of spore formation is that the spore formation leads to multiple number of offsprings. So millions of spores are released. So the chances for spreading out or perpetuation of species is more. And the chances for survival of that species is also very high because of the millions of spores. And each spore is covered by a special outer covering, a thick outer coat it has, and so it is protected from the surrounding environmental conditions. It is protected from moisture, it is protected from harsh sunlight and all other environmental conditions till it matures. So therefore, because of the huge number of spores and the fact that the spores are protected, spore formation is a very advantageous method. Also, the parents still survive, right? The parents are still there and they are able to produce new offsprings also. So that therefore spore formation is very advantageous. Next, let us move on to vegetative propagation. It is a type of asexual reproduction, okay? But sometimes vegetative propagation can be considered as a separate form of reproduction also, because asexual reproduction requires and involves certain specific parts of the body, parts of the organism, and it is a separate process. But vegetative propagation can be defined as a type of reproduction in which the non-reproductive parts are involved or the vegetative parts of the plant are involved. Okay, so vegetative propagation is exclusive to plants, and only the vegetative parts, that is root, stem and leaves of the plant, are involved in vegetative propagation. So vegetative propagation can be natural or of artificial type, and in artificial types we can see cutting, layering, grafting and tissue culture. These are the types that we are going to see. Natural vegetative propagation happens when plants grow and develop naturally without human interventions. So these are the naturally developed methods of vegetative propagation. Vegetative propagation takes place with the help of several structures, including roots, shoots and leaves. So in our day-to-day lives itself, we can see these forms of vegetative propagation. So vegetative plant structures that arise from plant stems include rhizomes, runners, bulbs, tubers and corms. Tubers can also stretch from roots. So the plantlets emerge from the plant leaves. So these are the different forms of natural vegetative propagation. The first one you can see is rhizomes. For rhizomes, the example is ginger. So ginger is an underground formation, right? But we usually think that it is a formation of the root. It is actually the formation of underground stem, right? So vegetative propagation may occur naturally through the development of rhizomes. Rhizomes are modified stems, they are not roots. Okay, so rhizomes are modified stems that typically grow horizontally along the surface or beneath the ground. So rhizomes are storage sites for growth substances such as proteins and starches, and they will grow into the new generation, right? As rhizomes extend, roots and shoots may arise from segments of the rhizome and develop into new plants. So these roots and shoots will develop into new plants, and therefore in ginger the rhizome actually helps in giving rise to the next generation. That is why so much of energy and proteins and starches are stored in the underground ginger, right? Certain grasses, lilies, irises and orchids propagate in this manner. Edible plant rhizomes include ginger and turmeric. So both ginger and turmeric are examples for rhizomes. Next, we'll move on to runners or stolon. Similar to rhizomes, they are similar in rhizomes in that they exhibit horizontal growth at or just below the soil's surface. Okay, so on the soil surface or just below the soil surface. Unlike rhizomes, they originate from existing stems. Okay, so they are not newly developing stems, but they originate from existing stems. As runners grow, they develop roots from buds located at nodes or their tips, and those roots will embed into the soil and become firm and give support to the stolons. Okay, intervals between nodes or internodes are more widely spaced in runners than in rhizomes. In rhizomes, the growth area is very less for the new plant, but in runners or stolons, long stems grow. Okay, so therefore internodes are widely spaced. New plants arise at nodes where shoots develop, and this type of propagation is seen in strawberry plants and in currants. Next would be tubers. All of you know that potato is an example for a stem tuber. So these are vegetative organs that may develop from stems or from roots. So stem tubers arise from rhizomes or runners that become swollen from storing nutrients. Okay, so in potato lots of nutrients are stored. That is for the next generation to develop. The upper surface of a tuber produces a new plant shoot system, stems and leaves, while the bottom surface produces a root system, example in potatoes and yams. So the shoot system will go up above the ground and it will start growing into a new plant, whereas the root system will help in embedding in the soil. Root tubers originate from roots that have been modified to store nutrients. These roots become enlarged and may give rise to a new plant, example sweet potatoes and dahlias. So potatoes and yams are example for stem tubers. Sweet potatoes and dahlias are example for root tubers. Then the next one would be bulbs. These are round swollen parts of the stem that are typically found underground. So from the diagram itself we can know garlic is an example for bulb, onion is also an example for bulb. Okay, so within these organs of vegetative propagation lies the central shoot of a new plant, and from that a new plant will develop. Okay, bulbs consist of a bud that is surrounded by layers of fleshy scale-like leaves. So in onions also you can see when you cut an onion, several layers are formed, right? So those layers are actually primitive leaves. They are actually called as scale leaves. They will provide nutrition to the newly growing plant. These leaves are a source of food storage and provide nourishment to the new plant. Examples of plants that develop from bulbs include onions, garlic, shallots, hyacinths, daffodils, lilies and tulips. Next, let us move on to corms. These are enlarged bulb-like underground stems. These vegetative structures store nutrients in fleshy, solid stems tissue and are typically externally surrounded by papery leaves. So that is the specialty of corms. They are externally surrounded by papery leaves. Due to their physical appearance, corms are commonly confused with bulbs. They almost look similar, but we can differentiate with the help of this papery leaves, right? The major difference is that corms contain solid tissue internally and bulbs have only layers of leaves. So this is one of the differences. Corms produce adventitious roots and possess buds that develop into new plant shoots. Okay, so plants that develop from corms include crocus, gladiolus, and taro. Next, let us move on to the artificial vegetative propagation. So artificial vegetative propagation happens when plants grow and develop naturally with human interventions. So humans carry out certain procedures in plants, certain methods in plants to make them grow artificially and to produce many number of offsprings. So human beings exploit the properties of plants of being vegetatively propagate, and there are several methods that can be applied for vegetative propagation in plants. So the first method we are going to see is cutting. A part of a plant, typically a stem or leaf, is cut off and planted. So this is commonly done in rose plants. Adventitious roots develop from the cuttings and a new plant will be formed. Okay, so cuttings are sometimes treated with hormones before being planted to induce root development. So hormones will help in assuring that roots will be developed, and they produce quick development of roots and growth of the plant also. Next would be grafting. You can see in the diagram different types of grafting. So it is simply a method of joining the vascular tissues of two different plants one over the other. So two different plants we are joining together in their stem region. Okay, so therefore that would lead to joining of the vascular tissues. Okay, so the plant that is being joined is called as the scion and the base plant is called as the stalk, and joining the scion and stalk together will lead to the growth of a new plant. So in grafting, a desired cutting or scion is attached to the stem of another plant that remains rooted in the ground. So the other plant is called as the stalk. The tissue systems of the cutting become grafted into or integrated with the tissue systems of the base plant over time. Okay, so after grafting, after some duration of time, the vascular tissues will become integrated and we can see properties of both the plants in a single plant. Okay, so the different types of grafting they have given examples are whip graft, splice graft, side graft, bark grafting, and saddle grafting. So grafting is a method of joining the stem of one plant with the stem of another plant, right? Next would be layering. Layering is a method which involves the bending of plant branches or stems so that they touch the ground. Okay, so many plant stems have the property that when they come in contact with soil, they will start growing roots. So this method is exploited in layering. The portions of branches or stems in contact with the ground are then covered with soil, and because of the presence of soil, adventitious roots will start to develop, right? So adventitious roots or roots that extend from structures other than the plant roots develop in the parts covered by soil, and the attached shoot, branch or stem with new roots is known as a layer. Okay, so you can see different types of layering in the diagrams: air layering, simple layering, tip layering, serpentine layering, trench layering, and mound or stool layering. Okay, so all methods of layering involve soil, and when the plants are attached to the soil, when they come in contact with soil, they start growing adventitious roots, and that leads to development of a new plant. So next is plantlets. These are vegetative structures that develop on some plant leaves. These miniature young plants arise from meristem tissue located along leaf margins. Upon maturity, plantlets develop roots and drop from leaves. Okay, so these are not considered as artificial methods, these are actually natural methods. Okay, so plantlets develop naturally in leaves of bryophyllum, and when these plantlets fall off, they enter into the soil, they start developing roots and they will grow as a new plant. Okay, so they then take root in the soil to form new plants. An example of plant that propagates in this manner is kalanchoe. Plantlets may also develop from the runners of certain plants such as spider plants. Okay, so plantlets or growth of miniature plants on leaves is also a type of vegetative propagation. The last type of vegetative propagation that we are going to see is tissue culture. So tissue culture involves the culturing of plant cells that may be taken from different parts of a parent plant and this method is completely done in the laboratory. Okay, so tissue culture depends on a particular phenomenon called as totipotency. The meaning of totipotency is the innate ability of any part of the plant to grow into a new plant. Okay, so the innate ability of any part of a plant to grow into a new plant is called totipotency, and that phenomenon or that concept is exploited over here by tissue culture. So the tissue is placed in a sterilized container and nurtured in a special medium until a mass of cells known as callus is formed. The callus is a mass of undifferentiated cells, and slowly those undifferentiated cells are developed in the laboratory. The callus is then cultured in a hormone-laden medium and eventually develops into plantlets. So when planted, these mature into fully grown plants. Okay, so the steps of tissue culture, you can see these are all carried out in the laboratory itself. So initially a small part of the plant is taken, either a part of a leaf or stem or root is taken, and then it is kept in the laboratory under certain conditions, so that it turns into a mass of undifferentiated tissue called callus, and slowly the callus multiplies and we separate out the callus into small portions, and the callus will start undergoing differentiation to develop roots and shoots. Okay, so after growth of roots and shoots, what would happen? They are all planted in the soil and they will start growing into new plants. So we are able to see that a whole plant can be grown from any small part of the plant. That is the speciality of tissue culture, and tissue culture is used for growing exotic plants, non-seasonal plants, and it is able to grow plants which are facing extinction also. So tissue culture has a lot of uses in our vegetative propagation. The second part of our chapter consists of the basics of sexual reproduction, which we will discuss in our next video. Thank you.